Disc 7.
By the mid-90s, with the initial mapping of the human genome accomplished and sequencing technologies fast developing, it was time to get down to the nitty-gritty of A's, T's, G's, and C's. Time to start sequencing. Sticking to the game plan outlined at the outset by our NAS committee, we would first attack an array of model organisms, bacteria to start with, and then on to more complicated creatures, with more complicated genomes. The lowly nematode worm, C. elegans, was the first big non-bacterial challenge, and as the joint achievement of John Sulston at Britain's Sanger Center and Bob Waterston at Washington University, it provided an excellent model of international collaboration. The worm sequence was published in December 1998, all 97 million base pairs of it. No bigger than a comma on this page, and comprising a fixed number of cells, just 959. The worm, nevertheless, has some 20,000 genes. At first sight, Sulston appeared ill-suited to a leadership role in big science. He had spent most of his professional life staring down a microscope in order to produce in astonishing detail a complete description, cell by cell, of the development of the worm. Bearded and avuncular, he's the son of a Church of England vicar, and also a lifelong socialist who believes passionately that business and the human genome should have nothing in common. Like Francis Collins, he is a motorcycle enthusiast. He used to commute on his 550cc machine from his home outside Cambridge to the Sanger Center until, just as the HGP was gathering speed, an accident left him severely injured and his bike, as he put it, little more than nuts and bolts. The Wellcome Trust, which was funding the Sanger Center, was horrified to learn that the project's scientific leader was taking his life in his hands every time he came to work. "After we'd invested all that money in this bloke," complained Bridget Ogilvy, then the Trust's director. Sulston's U.S. partner, Waterston, was an engineering major at Princeton and imported plenty of engineering savvy to the big sequencing center he ran at Washington University. Waterston has the capacity to extrapolate, to start small and finish big. Accompanying his daughter on a jog, he found he liked running and is now an accomplished marathon runner. During its first year of operation, his sequencing group produced just 40,000 base pairs of worm sequence. But within a few years, it was cranking out enormous amounts, and Waterston was one of the earliest to urge an all-out human sequencing effort. But even as those in the international HGP collaboration began to sequence model organisms, gearing up for the big one, the molecular biological equivalent of an earthquake shook the whole enterprise. Craig Venter and Tiger had been doing well. Having milked his cDNA gene discovery strategy for several years, Venter became interested in sequencing whole genomes. In this, too, he was persuaded by the superiority of his own approach. The HGP had been carefully mapping the location of the different chunks of DNA on the chromosomes before actually sequencing them. That way, you already knew that chunk A was adjacent to chunk B, and could look for overlaps between them when it came to knitting together the final sequence. Venter preferred a whole genome shotgun, WGS approach, in which there was no initial mapping. You simply broke the genome up into random chunks, sequenced them all, fed all the sequences into a computer, and relied upon the computer to put them all in the right order on the basis of overlaps, without benefit of any prior positional information. Venter and his team at Tiger showed that this brute force method could indeed work, at least for simple genomes. In 1995, they published the genome sequence of a bacterium, Haemophilus influenzae, using this method. It remained problematic, however. whether WGS would work for a large and complex genome like the human one. The problem is repeats, segments of the same sequence occurring in different places in the genome, which could in principle scupper a WGS sequencing attempt. These repeats might well mislead even the most sophisticated computer algorithm. If, for instance, a repeat occurs in chunks A and P, the computer could mistakenly situate A next to Q, rather than in its proper position, next to B. For its part, the HGP itself had discussed this scenario when it considered using a WGS approach. And based on careful calculations by Phil Green in Seattle, the consortium concluded that such an effort would likely be confounded by the human genome's massive amount of long-repeating sequences of junk DNA. In January 1998, Mike Hunkapiller of ABI, maker of automated sequencing machines, invited Venter to check out his newest model, the PRISM 3700. Venter was impressed, but nothing could have prepared him for what was to follow. Hunkapiller suggested that Venter form a new company, funded by ABI's parent company, PerkinElmer, to sequence the human genome. Venter had no misgivings about forsaking Tiger. Relations had long since soured with Hasseltine at HGS. And so he wasted no time in founding the firm that was later to be called Celera Genomics. The company motto? Speed matters. Discovery can't wait. The plan to sequence the entire human genome by WGS using 300 of Hunkapiller's machines and the single greatest concentration of computing power outside the Pentagon. The project would take 2 years and cost between $200 million and $500 million. The news broke just before the leaders of what would come to be called the public, as opposed to private Human Genome Project, were meeting at Cold Spring Harbor Laboratory. To put it mildly, the news was not well received. The Worldwide Public Project had already spent some $1.9 billion of public money, and now as the New York Times was spinning it, we might have nothing to show for the money except for the sequence of the mouse genome, while Venter waltzed off with the Holy Grail, the human genome. What was especially galling was Venter's flouting of what had come to be known as the Bermuda Principles. In 1996, at an HGP conference in Bermuda, a meeting Venter attended, the HGP had agreed that sequence data should be released as soon as it was generated. The genome sequence, we all concurred, should be public property. Now a renegade, Venter, had different ideas. He claimed he would defer releasing new sequence data for 3 months, selling licenses to pharmaceutical companies and any other parties seriously interested in